Assalamu alaikum. Our today's topic is compound microscope. You know that we have earlier done the simple microscope. In a simple microscope, we have a single convex lens, and in a compound microscope, we have two convex lenses, so that we have magnification is more than the simple microscope. So, what SLOs we are covering for the compound microscope? First is describe the working and uses of compound microscope, and other is just to draw a ray diagram of compound microscope and mention its magnifying power. So I'm sure you have used the compound microscope, you have seen the compound microscope in your school labs. Now, what are the uses of this compound microscope? A compound microscope is of great use in pathology labs, so as to identify the diseases. It is also used in the forensic laboratories to just detect and solve by drawing out human cells and examining them. Presence and absence of material and the presence of absence of metals can be identified using compound microscope. Students in schools and colleges benefit the use of microscope for conducting their academic experiments. It helps to see understand the microbial worlds of bacteria and viruses, and that which is otherwise invisible to the naked eye. Plant cells are examined, and the microorganisms thriving on it can be ascertained with the help of the compound microscope. Thereby, a compound microscope has proved to be crucial to the biologist. Now, the working of the compound microscope. For working, I would like you just to watch this video carefully so that the concepts, you can clear the concepts over here. So the first is the compound microscope.
Description. It consists of two converging lenses. The lens near the object is called objective, and the lens near the eye is called eyepiece.
Okay, now compound microscope, we have two lenses. You can see an objective and eyepiece. You can see that the objective is smaller than the eyepiece. So the focal length of objective is lesser, and the focal length of the eyepiece is more. So you can see that this is what the compound microscope is.
The lenses are fitted at the end of a metal tube. The distance of the object from the objective can be adjusted by means of a rock and pinion arrangement. Works. The object OJ to be magnified is placed just outside the principal focus of the objective so that its real image is formed on the other side of the objective.
Okay, now you can see that uh, this object is placed just outside the focal length, just to have a real inverted and magnified image of the from this objective lens. So just see that how the ray diagrams are made from this.
The image forms I1G1 beyond 2F0, where F0 is the focal length of the objective. The image due to the objective acts as the object for the eyepiece. The position of the eyepiece is so adjusted to form the final image at the near point. The final image is virtual, inverted, and magnified. It is IG. To find the magnifying.
Okay, now, so you can see that how these are traced. You can see that this is the working. The object is placed beyond the focus. The first ray that is coming out parallel to the principal focus, after passing through the focus, uh, and the second ray is that is passing from the curvature, passes unrefracted, and they make an image. The distance between the two lenses can be increased and decreased, so this image is adjusted in such a way that this image serve as an object for the other lens. So you can see that this is an object for this lens. Now the first ray is drawn parallel to the principal axis, and it has to be passed from the focus, and the second ray that is passing from the curvature will passes unrefracted, and there is a diverging rays on this side, and they converges over here. If I produce these rays over here, they converges at a point, and the point where it converges, a magnified image of this object, this fly, is formed. So this is the working of a compound microscope. Okay, now, so just we are going to see that in this diagram, this is the object that is between FE is the object focal length of the objective, and this is the focal length of the eyepiece. Now you can see that object is placed beyond the focus. So first ray, and after plotting the ray diagram, I can see that there over here the image is formed, and the image is a real inverted and a big enlarged image. Now this image is adjusted in such a way that it lies within the focal length of your eyepiece. So when the object is in within the focal length of the eyepiece, we will get a virtual erect and inverted image. So you can see that a little object is how much magnified by these two lenses. So this is the working of a simple microscope. So uh, and how it is giving a very high magnification. So what is the magnifying formula for this? You can see magnifying formula is equal to m1, that is the magnification of objective, multiply by m2, that is the magnification of eyepiece. Now first the objective lens, we have linear magnification, that is we can say that is q by p, or we can say d by p. So over here the magnification of m1, that is the linear magnification, is L by f naught, where L represent the distance between the objective and the eyepiece, and f naught is the focal length of the objective, and m2 is used in such a way so it gives a final image as a virtual erect and highly magnified image. So for the from the from the simple microscope formula, 1 plus d by fp. So d is the least distance of distinct vision in which the final image is formed, and fe is the focal length of your eyepiece. Okay, now the ray diagram of compound microscope. We have done the ray diagram over in the previously we have seen the working, and you can see the object is over here, ray parallel to the principal axis after refraction passing from the focus. The ray that is passing from the focus passes unrefracted. These two meet over here, so and real inverted and enlarged image is formed over here. Now eyepiece is we are using the eyepiece of a larger focal length. So this uh, image of this lens is adjusted in such a way that it serves as an object for this lens, and it should be within the focal length of this lens. Now you can see FE is the focal length of the eyepiece, and this is object is within the focal length of this eyepiece. Now again I'm going to draw the ray diagram. You see that the ray that is parallel to the principal axis after refraction passes from the focus. The ray that is passing from the optical center passes unrefracted. Over here, these two are the divergent rays, and they will never meet. So I have to produce them back, and produce them back, they meet over here, and you can see a highly erect and magnified erect and image is formed at the least distance of distinct vision. So this is the ray diagram for the compound microscope.